If you spend any time in biohacking communities, recovery forums, or sports medicine discussions, you have almost certainly encountered BPC-157. The claims surrounding it are remarkable. Tendon healing, gut repair, systemic inflammation control, accelerated tissue recovery, wound healing, neural protection. The list, depending on where you look, seems to grow indefinitely. And so does the skepticism. Critics point to the near total absence of controlled human data, the concentration of research from a single laboratory group, the fact that no regulatory agency anywhere in the world has approved BPC-157 for human use, the gray market supply chains, the quality risks. Both of those positions, the enthusiasm and the skepticism, exist because the actual science is genuinely complicated. The biology is interesting. The human evidence is thin. Both of those things are true at the same time. This video is not here to sell you on BPC-157. It is also not here to dismiss it. It is here to give you the tools to understand what the evidence actually shows and what it does not, so you can evaluate any claim you encounter with something more useful than hope or skepticism alone. We are going to use one filter throughout this entire video. For every claim, what tissue, what route, what endpoint, and what level of proof? Keep those 4 questions in mind. They will do a lot of work. BPC-157 explained what it is, how it works, and what the evidence actually shows. Let's start with the basics. What is BPC-157? BPC stands for Body Protection Compound. The 157 is an identifier from the original research literature. It does not mean the molecule contains 157 amino acids. That is one of the most common misconceptions about this peptide. BPC-157 is 15 amino acids long. BPC-157 equals Body Protection Compound. 15 amino acids long, not 157. 157 is a research identifier, not a length. In formal research and commercial peptide contexts, BPC-157 is described as a synthesized pentadecapeptide. Pentadeca meaning 15, so a 15 amino acid chain, synthetically produced. Its origins trace back to a gastric protection research program. The original literature described BPC-157 as related to protective compounds identified in human gastric juice. The idea from the beginning was stomach cytoprotection, protecting the cells of the stomach lining from injury. You may also encounter BPC-157 under other names in the literature and the commercial space: Bepicin, PL10, PLD116, PL14736, PCO02. These labels are not always used in identical contexts, but they generally refer to the same family of BPC-related research peptides. One property that drew early research interest is BPC-157's reported stability. Most peptides break down quickly when exposed to the acidic environment of the stomach, which makes oral administration essentially useless for most of them. BPC-157, by contrast, has been described in the literature as relatively stable in gastric juice. That stability is part of why researchers explored it in oral and gastrointestinal studies from early on. One more thing to establish clearly before we go further: BPC-157 is not an FDA-approved drug for any indication. Not in the United States, not anywhere in the world. We will get into the current regulatory picture in detail later in this video, because the situation as of July 2026 is genuinely significant and worth understanding precisely. But for now, the foundation. A 15 amino acid synthetic peptide, originally from gastric protection research, with demonstrated stability in gastric juice and no current regulatory approval for human therapeutic use anywhere on the planet. To understand why BPC-157 attracts attention and why the proposed mechanisms make biological sense, you need to understand the specific problem it is most often applied to. Tendons and ligaments are slow healers. That is not an opinion. It is a reflection of their tissue biology. These are dense, collagen-rich structures, And compared to muscle or skin, they have relatively limited blood supply. Less blood supply means fewer nutrients reaching the injury site, fewer repair cells being delivered, less oxygen, less of the signaling molecules that coordinate healing. All of that slows recovery and makes it less predictable. Tissue healing happens in phases. The first phase is inflammation. And it is worth noting that inflammation here is not automatically the enemy. It is part of the process. It clears damage and triggers the signals that initiate repair. Then repair cells, fibroblasts and related connective tissue cells, have to migrate into the injury site. If they cannot get there, they cannot build the repair matrix. Once they arrive, they begin laying down collagen. Early collagen is disorganized, more of a patch than a functioning structure. It closes the gap before it has the strength or alignment of healthy tissue. That alignment comes later through mechanical loading and the remodeling phase. This is why structural healing takes longer than symptom relief, and it is why that distinction matters enormously when evaluating any claim about BPC-157. If someone says their pain improved, that matters clinically. Pain is a real and meaningful endpoint. But improved pain is not the same thing as a tear closing, not the same as collagen reorganizing, not the same as load to failure strength returning to baseline. Those are different endpoints, and a complete picture of healing requires evidence at more than just the symptom level. Pain relief doesn't equal structural healing. Keep this distinction in mind for every claim you evaluate. Real tissue healing touches on one or more of 5 things: blood supply, repair cell behavior, the extracellular matrix structure, inflammation control, and time. BPC-157 gets research attention because the preclinical literature reports activity across several of these, not just one. That breadth is what makes the mechanism worth taking seriously. It is also what makes clean evaluation harder. BPC-157 is not typically described as acting through one clean receptor-ligand pathway. Researchers describe it more as a cluster of overlapping effects involving blood vessel behavior, tissue protection signals, and repair cascades operating simultaneously. Let's take each major pillar separately. The first is the vascular theme. BPC-157 appears repeatedly in research examining how it affects endothelial cells, the cells lining blood vessels, nitric oxide signaling, and new vessel formation. A 2017 mechanism paper focused on a receptor called VEGFR2, vascular endothelial growth factor receptor 2. This receptor is part of the system tissues use to build and organize blood vessels. It is a central player in angiogenesis, the formation of new blood vessels. The model here is not a simple textbook plug-in. BPC-157 was not simply described as a direct ligand for VEGFR2. What the study reported was increased VEGFR2 expression and receptor internalization, which then connected to downstream signaling cascades. VEGFR2 activation, AKT pathway, eNOS, nitric oxide, vascular effects. One downstream pathway from active VEGFR2 signaling is AKT, a molecule involved in cell survival, growth, and certain repair signals. AKT can in turn activate eNOS, endothelial nitric oxide synthase. That is the enzyme endothelial cells use to produce nitric oxide. Nitric oxide is one of the primary regulators of vascular tone and endothelial function. It relaxes blood vessels, it influences blood flow, and it can support endothelial cell movement, which matters for repair processes. A 2020 paper in Scientific Reports added another layer, the SRC-caveolin-1 eNOS pathway, as an additional route through which BPC-157 could activate eNOS signaling inside endothelial cells. In that study, the nitric oxide dependency was tested directly. When researchers blocked nitric oxide signaling, the vasodilation response was reduced. So the more accurate statement is not simply that BPC-157 boosts nitric oxide, It is that BPC-157 modulates nitric oxide-related pathways in preclinical systems, particularly around endothelial function and repair environments. More accurate, BPC-157 modulates nitric oxide-related pathways in preclinical systems, cells, isolated vessels, and rodent models. In rodent vascular studies, researchers also describe collateral pathway activation, where smaller blood vessels develop to help route around a blocked or damaged vessel. That concept is consistent with the vascular repair model. It is also, again, animal evidence. And this is the pattern you will see throughout this video. The biological mechanism of BPC-157 is plausible, and the preclinical signal is real. The consistent caveat is where the evidence has been generated: cells, isolated vessels, rodent models. Still, if BPC-157 genuinely modifies endothelial behavior and the blood supply environment around damaged tissue, there is a logical pathway to influence in tissues where blood supply is the main bottleneck to healing: tendons, ligaments, gut lining. That is the first pillar of the proposed mechanism. BPC-157 may change the repair environment by changing vascular behavior. The second pillar moves from blood vessels to the cells that actually build the repair. In tendons and ligaments, repair cells have to do several things in sequence. They have to detect the injury, they have to migrate toward it, they have to survive the inflammatory stress environment they are entering. They have to attach to the damaged matrix, and then they have to organize the new collagen they are laying down. Each one of those steps is a potential point of failure, and each one is where BPC-157 research has looked for effects. A 2011 study in the Journal of Applied Physiology used tendon explants and isolated tendon fibroblasts, the primary repair cells in connective tissue. What the researchers found was that BPC-157 accelerated outgrowth from tendon explants, meaning repair cells moved out from the tissue more readily in that experimental setup. The same study reported increased fibroblast migration. Migration is not an abstract laboratory endpoint. In a real injury, cells have to physically travel into the damaged area before they can do anything useful. If migration is impaired, the repair process stalls at its second phase. The same study tested oxidative stress conditions using hydrogen peroxide, simulating the inflammatory stress environment an injured tissue creates. BPC-157 did not simply cause fibroblasts to multiply in every condition. What it did do was improve cell survival under that stress. That distinction matters. A compound that helps cells survive stress in an injury environment is not the same as one that simply accelerates proliferation indiscriminately. The study also reported changes in F-actin formation, the internal scaffolding a cell reorganizes when it needs to spread, pull, and move. And BPC-157 increased phosphorylation of two proteins, FAK and paxillin, that are involved in focal adhesions. Those are the contact points where a cell grabs onto its surrounding matrix to pull itself forward. Phosphorylation, for context, is the addition of a phosphate group to a molecule. It is one of the primary ways cells switch signaling pathways on. This helps explain why the tendon repair story here is not simply a more collagen question. It involves cells migrating into the injury, surviving the stress, attaching to the matrix, and organizing the tissue. BPC-157 appears to affect multiple points in that sequence in preclinical tendon models. A separate finding in the fibroblast literature is also worth noting carefully. One study found that BPC-157 increased growth hormone receptor expression in tendon fibroblasts, and when growth hormone was added to BPC-157-treated cells, downstream JAK2 signaling and proliferation-related measures increased. That finding requires precision in how it is interpreted. Growth hormone receptor upregulation in tendon fibroblasts does not mean BPC-157 is growth hormone. It does not establish that combining BPC-157 with growth hormone or growth hormone secretagogues is safe or effective. In fact, combining compounds in that way introduces significant confounders that make it harder to attribute any outcome to any individual ingredient. GH receptor upregulation in fibroblasts does not equal BPC-157 is growth hormone, does not equal stacking is safe or effective. It is a narrow in vitro finding, not a human treatment claim. The honest summary of Pillar 2? In preclinical tendon models, BPC-157 appears to influence repair cell migration, cell survival under stress, adhesion signaling, and possibly responsiveness to growth hormone-related pathways. All of that is in cells and animal models. None of it is proven in human tissue under controlled conditions. The third pillar goes back to the origin of BPC-157 research, the gastrointestinal tract. BPC-157 comes out of a gastric cytoprotection research tradition. Cytoprotection? Protecting cells from injury and maintaining tissue integrity. In the early literature, that meant the stomach lining and gut mucosa specifically. Think of barrier tissues as living walls. When those walls are intact, they separate the contents of the gut from the bloodstream and the surrounding tissue. When they break down, through injury, inflammation, medications like NSAIDs, alcohol exposure, or conditions like ulcerative colitis, the consequences include pain, inflammation, poor healing, and in severe cases, structural damage to the gut architecture. Animal studies and reviews describe BPC-157 as offering protection against various forms of gastric injury, including injury from NSAIDs and alcohol. The proposed mechanism is not acid suppression. It is closer to a tissue protection factor, something that helps maintain barrier integrity rather than simply reducing acid output. One idea that appears repeatedly in the literature is that epithelial protection and endothelial protection are connected. The cells lining the blood vessels beneath the gut surface influence whether the surface barrier itself can recover from injury. That connection links the vascular pillar and the gut pillar together. They are not entirely separate effects. The gastrointestinal animal literature spans a remarkable range of injury models. Ulcers, colitis, fistulas, ischemia-reperfusion injury, where cutting off oxygen to tissue and then restoring it paradoxically damages that tissue. Anastomosis healing, repair after surgical reconnection of the bowel. Gut models tested: ulcers, colitis, fistulas, ischemia-reperfusion injury, anastomosis healing. Very different injuries. Shared theme: barrier damage and repair. With the three mechanism pillars established, let's look at what the animal evidence actually shows across the literature. The body of preclinical work on BPC-157 is genuinely large. Across published studies, researchers have reported effects in skin wound healing, gastrointestinal injury, tendon repair, ligament healing, muscle recovery, vascular recovery models, bone healing, nerve repair, and spinal cord injury models. That breadth is part of why BPC-157 generates so much discussion. When a compound appears across that many tissue types, it suggests either a very fundamental biological mechanism or a literature with other characteristics we should examine carefully. A few specific studies are worth grounding this in concretely. A 1997 wound healing study in rats tested BPC-157 across multiple models: skin incisional wounds, colon anastomoses, and a synthetic sponge angiogenesis model. The study assessed granulation tissue formation, collagen quality, angiogenesis, and tensile strength, meaningful tissue repair endpoints, not just symptom measures. Results favored BPC-157-treated animals across multiple endpoints. A 2003 rat Achilles tendon study used a severe model, complete transection of the Achilles tendon, leaving a full gap defect. BPC-157-treated animals showed improved functional recovery, higher load-related mechanical measures, better microscopic tissue formation, and smaller tendon defects over time. That is functional and structural evidence in an animal model, stronger than symptom reporting alone. A 2010 medial collateral ligament study in rats with surgically transected MCLs showed similar results across functional, biomechanical, macroscopic, and microscopic measures during the healing period. One detail researchers often note about rodent studies is route flexibility. Studies report benefit with intraperitoneal injection, oral administration in drinking water, local application, and topical routes. That route flexibility gives researchers reason to investigate translation to different delivery methods. It does not, however, answer how those routes translate to human use. Now, here is something important about this literature that an honest evaluation cannot skip. A December 2025 scoping review noted that across published BPC-157 studies, all reported positive effects, every single one. All published BPC-157 studies report positive effects. This itself is a signal worth examining. In science, that kind of uniformly positive publication record is unusual. It raises the question of publication bias, whether negative or null results exist but have not made it into the published literature. Negative results are harder to publish and less likely to be submitted. If researchers ran experiments that found no effect of BPC-157 and those experiments went unpublished, the visible literature would look more positive than the full picture actually is. Additionally, a substantial portion of BPC-157 research has come from a relatively concentrated group of research laboratories. Independent replication across multiple groups in different institutions, one of the basic quality signals in science, is more limited than the volume of published studies might suggest. Breadth of animal models is interesting evidence. It is not certainty, and the translation from a rodent Achilles tendon to a human athlete involves questions that animal studies cannot answer. Correct human dosing, appropriate imaging endpoints, return to sport protocol, how the immune system responds chronically. None of those have been studied systematically. This is the section that requires the most precision, because the gap between what is claimed for BPC-157 and what has been tested in humans is one of the largest in the peptide space. As of mid-2026, 3 human studies on BPC-157 have been published. All 3 were conducted by the same research group. All 3 are small pilot studies. None has a placebo control group. Let's go through each one. The 2021 knee pain study looked at 16 patients with chronic knee pain receiving intra-articular BPC-157 injections. 14 of the 16 reported significant pain relief at 6 to 12-month follow-up. That is a positive signal. The limitation is the absence of a placebo control. Pain is a subjective measure and highly susceptible to placebo effects. Without a group receiving an identical appearing injection of an inert substance, there is no way to separate the drug's effect from participant expectations, the natural trajectory of the condition, or the act of receiving an intervention itself. That is not a minor methodological gap. It is the fundamental question the study design cannot answer. 2024 Interstitial Cystitis Pilot. 12 participants, women with moderate to severe bladder pain syndrome. Intravesicular injection into the bladder. 10 of 12 reported complete symptom resolution. Remaining 2 reported approximately 80% symptom improvement. The 2024 bladder pain pilot is the most interesting human signal in the BPC-157 literature. 12 participants with moderate to severe interstitial cystitis, a chronic inflammatory bladder condition, single intravesicular injection. 10 of 12 reported complete symptom resolution. 2 reported approximately 80% improvement. That is a striking result, and it earns the characterization of a signal worth following, specifically because interstitial cystitis involves the kind of barrier tissue inflammation and microvascular dysfunction that BPC-157's proposed mechanism is actually designed to address. The biological logic is coherent. The honest framing? 12 people, no placebo group, single injection. That is not a treatment established by evidence. It is a direction worth investigating properly. The 2025 IV safety pilot had 2 participants. 2. No adverse events were reported, which is noted, but 2 participants is nowhere near enough to characterize the safety profile of intravenous BPC-157 in humans. A safety signal, positive or negative, from 2 people cannot be generalized. There is also a canceled study worth understanding. A Phase 1 pharmacokinetics trial registered as PCO-02, one of the alternate BPC-157 compound names, enrolled approximately 42 healthy volunteers and aimed to establish the first rigorous human safety and pharmacokinetics data. In 2016, results were withdrawn. The study was canceled. No data was ever published. That cancellation matters for a specific reason. Pharmacokinetics data, how the drug absorbs, distributes, metabolizes, and is eliminated in humans, is the foundation for rational dosing. Without it, any dose being used today in humans is extrapolated from animal studies, not based on actual human metabolism data. Human dosing today, extrapolated from animal data. No published human pharmacokinetic study exists. There is one actively recruiting human trial worth watching, a Phase 2 randomized, double-blind, placebo-controlled study of BPC-157 for acute Grade 2 hamstring strain, confirmed by MRI, registered on clinicaltrials.gov as NCT-074-37547. It is the most rigorously designed human study of BPC-157 ever attempted. It has MRI-confirmed injury endpoints, return-to-play follow-up, and a proper placebo arm. Results, when they emerge, will be genuinely informative.
Phase 2 hamstring trial, NCT-074-37547. MRI-confirmed endpoints, placebo-controlled, currently recruiting, first rigorous human efficacy test for BPC-157. The honest summary of the human evidence: it exists, it carries some biological plausibility, and it is too small and methodologically limited to support confident conclusions about efficacy in any indication. The picture may look different once larger, controlled trials produce results. Right now, the preclinical map is large, and the human evidence is a series of very small dots.
The regulatory situation around BPC-157 has moved more in the past 6 months than in the previous 6 years. Understanding it precisely matters. Here is the current picture. BPC-157 is not an FDA-approved drug for any indication. That has not changed. 2019, first oral GLP-1 approved. Background context for regulatory environment. 2022, WADA adds BPC-157 to prohibited list, S0 category. 2023, FDA moves BPC-157 to Category 2 bulk drug substances list, cannot be legally compounded. April 2026, FDA removes BPC-157 from Category 2 list after nominations withdrawn. May 2026, BPC-157 absent from updated Category 2 document. July 23rd to 24th, FDA Pharmacy Compounding Advisory Committee, PCAC, scheduled meeting. In April 2026, the FDA removed BPC-157 from its Category 2 bulk drug substances list. Category 2 was the classification that explicitly prohibited compounding pharmacies from preparing BPC-157. Its removal from that list means the explicit prohibition was lifted. That is not the same as approval, not even close. Removed from Category 2 does not equal FDA approved. It means no longer explicitly prohibited for compounding pharmacies. It does not mean safe, effective, or legal to prescribe freely. The next step in the formal process is the FDA Pharmacy Compounding Advisory Committee meeting scheduled for July 23rd and 24th, 2026, which, depending on when you are watching this, may have just happened or is imminent. The committee is reviewing whether BPC-157, in both its freebase and acetate forms, should be added to the Section 503A bulk drug substances list. Getting on that list would give licensed compounding pharmacies a formal legal pathway to prepare BPC-157 for patients with a physician's prescription. The proposed use being reviewed is ulcerative colitis. The committee is also reviewing TB500, KPV, MOTS-C, Samax, Epitalon, and DSIP at the same two-day meeting. A positive vote from the committee does not automatically mean approval.
The FDA is not bound by advisory committee recommendations, though it has generally followed them. A positive vote would need to be followed by a formal FDA determination before any change in legal compounding status takes effect. And regardless of what happens at that meeting, BPC-157 is still not FDA approved, will not become FDA approved through a PCAC vote, and any BPC-157 currently available through commercial channels is operating outside the formal pharmaceutical supply chain. For athletes specifically, BPC-157 is prohibited by WADA under the S0 non-approved substances category, which covers any pharmacological substance with no current approval by a governmental health authority for human therapeutic use. It is prohibited at all times, in and out of competition. Anti-doping laboratories can detect it. The NFL and UFC also prohibit it explicitly. For any athlete subject to anti-doping testing, this is a straightforward calculation.
This is the question that comes up most often and deserves a direct, careful answer. BPC-157 is discussed in repair and recovery contexts partly because of its angiogenesis activity. It appears to promote new blood vessel formation, and tumors use blood vessel formation too, so the concern is understandable. Left pathway: injured tendon plus BPC-157 plus angiogenesis plus new vessels plus healing tissue. Right pathway: pre-existing tumor plus angiogenesis plus new vessels plus accelerated tumor growth. The honest answer is not that BPC-157 definitively causes cancer. The evidence for that claim does not exist. A more accurate framing of the concern is this: if BPC-157 modulates angiogenesis systemically, and if that modulation reaches precancerous cells or an existing tumor that has not yet been detected, it is biologically plausible that increased blood vessel growth in that context could support more rapid progression. The key word is plausible, not proven, not observed in human populations. Plausible based on the known biology of angiogenesis and tumor development. The questions that remain genuinely unanswered are: does the risk change with dose and duration? Does it depend on the injury state and location? Would an Achilles tendon injury produce localized vascular changes or systemic ones? Does age play a role in how the body responds to angiogenic signaling from this compound? Those questions are not answered in the preclinical literature. They have not been studied in humans. They are real unknowns, not confirmed dangers, but also not dismissed risks. Not BPC-157 causes cancer. More accurate, angiogenic activity in the context of undetected tumors is a plausible theoretical concern. Status: unknown, not studied in humans. This is one of the areas where the absence of long-term human data is felt most acutely. Without chronic exposure studies in humans, we simply do not know how systemic or localized the vascular effects of BPC-157 are, or whether they create meaningful cancer risk at the doses and durations people are actually using. There is one more risk category that is separate from BPC-157's pharmacology entirely, and for many people using it right now, it may actually be the most immediately relevant risk. When BPC-157 is obtained through gray market channels, which, given its regulatory status, is the only way most people are currently accessing it, the questions you need to be asking are not just about the compound's biology. They are about what is actually in the vial. Identity: is it really BPC-157? Purity: what else is in it? Concentration: is the labeled rate? Sterility: has it been properly prepared? Endotoxin: is there bacterial contamination? Endotoxin contamination is worth understanding specifically. Endotoxins, compounds like lipopolysaccharides produced during bacterial fermentation, can remain in a vial even after the bacteria that produced them are gone. When injected, they can trigger significant inflammatory responses. In sufficient amounts, they can cause sepsis. The BPC-157 in a vial may be entirely legitimate, but the preparation process, if it was not conducted under pharmaceutical-grade sterility controls, can introduce hazards that have nothing to do with BPC-157 itself. This is also why stacking compounds, Combining BPC-157 with TB500 or with growth hormone secretagogues or with PRP creates an evidence problem as well as a safety problem. If someone uses a combination and has a response, positive or negative, attributing that outcome to any one component becomes significantly harder. The confounders multiply with every compound added. For every BPC-157 claim, ask 4 questions. What tissue? Is this the same tissue the evidence comes from? What route? Oral, subcutaneous, intra-articular, IV? Evidence differs by route. What endpoint? Pain relief or structural healing? Symptom or function? What proof? Cell study, animal model, or controlled human trial? The preclinical repair signals around BPC-157 are strong enough that researchers and clinicians continue returning to this compound. Across the literature, repeated effects appear involving blood vessel behavior, repair cell movement, barrier tissue integrity, and inflammatory control. There is clearly something biologically interesting here. The human gap is still very large. There is no published randomized controlled trial, no FDA approval, no established safe and effective dose for humans derived from human pharmacokinetic data, and a regulatory status that, as of mid-2026, is in active transition rather than settled. When you hear a claim about BPC-157, run it through that 4-question filter. What tissue? What route? What endpoint? What proof? If the answers are vague, the claim is probably outpacing the data. If the answers are specific and grounded in actual study designs, you have something worth thinking about. BPC-157 has the biology that makes definitive answers worth pursuing. The science has not caught up to that yet. When it does, and the hamstring trial and the July 2026 FDA review are both steps in that direction, the picture will be clearer. Until then, interesting mechanism, promising preclinical signal, thin human evidence, regulatory situation in flux. That is where it actually stands. If this breakdown helped you evaluate BPC-157 more clearly, share it. Most discussions of this compound go to one extreme or the other. People who want the nuance need to be able to find it. Subscribe to What the Health. New video every week.